Good evening to everybody. Good evening to all. Okay. Today I would like to explain digestion and absorption. And absorption. Okay. Digestion and absorption. Okay. In that we can discuss the 2 marks questions. We can discuss the 2 marks questions. First 2 marks question. Give the dental formula of human beings. Give dental of human beings. Give the dental formula of human beings. What is a dental formula? So, dental formula we can give arrangement of different types of teeth. Arrangement of different types of teeth. Right. This is the first question. Arrangement of different types of teeth. Different types of teeth on each of the both jaws. On teeth in each half of the jaws in order. One is incisors. I means incisors. C means canines. C means canines. And third one, PM, premolars and molars. Premolars and molars. What is the dental formula in adult human beings? In adult human beings, in adult human being, two by two, one by one, and 2 by 2, 3 by 3. 2 by 2 is first one is the incisors, canines, premolars and molars. That is about the jaws. So first here if they are asked what is the dental formula of the human beings. So the different types of teeth are arranged. Generally there are 4 types of teeth arranged in each half of the jaws. They are 1. Incisors, canines, premolars and molars. In adult human beings, dental formula is 2 by 2. 2 by 2 means upper jaw is 2 incisors, lower jaw 2 incisors. And canines, upper jaw 1 and lower jaw 1. Premolars, upper jaw 2, lower jaw 2. And molars, upper jaw 3, half of the upper jaw and half of the lower jaw is 3. That is about the the dental formula. Okay, then what is the importance of the bile juice? Bile juice contains no digestive enzymes. Second question. Bile juice contain no digestive enzymes. No digestive enzymes. It is important for digestion. It is important for digestion. Important for digestion. How? How it is important? So bile juice does not contain digestive enzymes. Answer is bile juice does not contains bile juice does not contains digestive enzymes. Digestive enzymes. Digestive enzymes, but it contains bile salts. But it contains bile salts. Bile salts such as glycocolate, glycocolate, and tarocolate, of tarocolate of sodium and potassium. Sodium and potassium. Sodium and potassium. Okay, then and they help, these bile salts, they help in emulsification of, they help in emulsification of fats. Emulsification is means breakdown of fats into a very small micelles. Fats into very small molecules or micelles. That is about the digestive enzymes, bile juice. And the third one is question number 3. What would happen if HCl were not secreted in the stomach? What would happen? HCl, what would happen if HCl were not secreted in the stomach? In the stomach, what happens in the stomach? If the stomach fails to secrete HCl, if stomach fails to secrete, if stomach fails to secrete HCl, the conversion of inactive pepsinogen into active pepsin, conversion of inactive pepsinogen into pepsin. 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 And prorenin into prorenin into renin. Prorenin into renin does not takes place. Does not takes place. So digestion of proteins and a milk protein. So digestion of proteins and milk. Protein in the proteins and milk. Milk protein. Milk protein in stomach is impaired. In stomach. Impaired or not digested. Not digested. So, what happened if the HCl were not secreted in a stomach? The answer is, in stomach, if the stomach is fails to secrete HCl, conversion of inactive enzyme, that is pepsinogen into pepsin, prorenin into renin, does not take place. Prorenin into renin does not takes place. So digestion of protein and a milk protein in stomach impaired or not digested. That is about the HCl importance. And the next one is the explain the term thecodont and diffeodont or diffeodont. Fourth one is explain the terms thecodont and diffeodont. Diffeodont dentition. Diffeodont dentitions. First we can take the answer thecodont. What is the thecodont? In thecodont, if teeth are embedded in the sockets of jawbone, if teeth are embedded in the jaw socket, the teeth are embedded in the jaw sockets of bone. Such dentition is called thecodont. Such dentition is called thecodont. And diffeodont. What is a diffeodont? In diffeodont teeth, if two sets of teeth, tooth, that is milk set and a permanent set, milk set and permanent set milk set and permanent set are formed in the lifetime. Formed in the lifetime. Formed in the lifetime. Such type of dentition is called. Such type dentition is called diffudent. Such type of dentition is called diffudent. Right? And then, so in the terms, one is the thecodont and second one is diffudent dentition. Thecodont, if the teeth are embedded in jaw sockets or sockets of the jawbone, such dentition is called thecodont. Second one is diffudent. If two sets of teeth, that is milk teeth and permanent teeth, or milk set and permanent set, formed in the lifetime, such type dentition is called the diffudent teeth. Next, what is chyme? Next question is, what is chyme? What is chyme? So chyme is a partly digested acidic food. Chyme is partly digested acidic food formed in stomach. Formed in stomach. Formed in the stomach. It is formed by mixing of ingested food. It is formed by mixing of ingested food. Ingested food with gastric juice. Gastric juice. With gastric juice by churning movements. By churning or thorough mixing. Thorough movements. Thorough movements of stomach. Thorough movement of the stomach. That is the chyme. What is chyme? Chyme is partly digested acidic food formed in the stomach. It is formed by the mixing of ingested food with the gastric juice by churning movement of stomach. Next, name the different types of salivary glands. Name the different types of salivary glands. Different types of salivary glands in their locations in the human body. Glands and their locations and their locations in the human body. In the human body. Answer: the glands. Three pairs of salivary glands present in human body. Three pairs of salivary glands present in human body, human beings. Human beings. They are parotid glands. First one is parotid. Parotid glands. First one is parotid glands present below the pinna. Present below the pinna or inner surface of cheeks. Inner surface of cheeks. Second one is submaxillary or submandibular glands. Submaxillary or submandibular. Submaxillary or submandibular glands. Present on the surface of tongue. Present on the at the angles of lower jaw. Present at angles of lower jaw of lower jaw. Angles of lower jaw. And third pair is sublingual glands. Sublingual glands present below the tongue. Sublingual glands present below the tongue. That is about the different types of salivary glands. Name the different types. Next question. Name the different types of papillae. Types of papillae present on tongue of man. Present on the tongue of man. 1. So, in human beings, tongue bears 3 types of papillae. In human beings, tongue bears 3 types, not pairs. 3 types of papillae. First one is fungiform papillae. Fungiform papillae. Fungiform papillae present at anterior margin of present at the anterior margin of tongue. And the second one is filiform. Filiform papillae. Filiform papillae. Present on surface of tongue. On surface of tongue. Third one. Circumvallate. Circumvallate papillae. Circumvallate papillae. Present on posterior surface or base of the tongue. Posterior surface or base of tongue. Base of tongue. Suppose this is the tongue, for example. Here they are fungiform. Fungiform papillae, and all these are filiform, and here are circumvallate. Circumvallate papillae. Okay, these are the three types. This is the surface of the tongue. Margin, anterior margin of the tongue, and posterior base of the tongue. That is about that. And next one is what is the hardest substance? What is hardest substance in the human body? What is the hardest substance in the human body? What is its origin? What is its origin? Answer: the hardest substance in the human body is enamel. The hardest part in the human body is enamel, that covers the crown of tooth. That covers crown of tooth. It's secreted by amyloblast of ectodermal amyloblast cells of ectodermal origin. Ectodermal origin. The next one is the or digestive substance in the body, human body. Next one is 4 marks questions in this. Describe the process of digestion of proteins in SAQ questions. Short answer question. First question is, in this chapter, the diagram, important diagram is draw a neat label diagram of LS of tooth. Draw a neat label diagram of LS of tooth. It is also very important. They will ask. Next one. Describe the process of digestion, digestion of proteins. Digestion of proteins in the stomach. Digestion of proteins in the stomach. That is the question. The answer is first point. The food bolus swallowed into the stomach. The food bolus the food bolus swallowed into stomach mixed thoroughly acidic mixed thoroughly mixed thoroughly with acidic gastric juice. With acidic gastric juice. Gastric juice of stomach. Gastric juice of stomach by churning movements of its muscular wall. By churning movements of its muscular wall. By its muscular wall. The partly digested acidic food. Partly digested acidic food. The partly digested acidic food formed in stomach is called formed in the stomach. In the stomach is called chyme. Mucus and bicarbonates. The mucus and bicarbonates present in the gastric juice play an important role in lubrication play an important role in the lubrication and protection of lubrication and protection lubrication and protection of mucosal epithelium mucosal epithelium from excoriation excoriation by highly concentrated HCl by highly concentrated HCl. If the bicarbonates and mucus are not present in the stomach mucosal epithelium is damaged. That process is called excoriation. So excoriation is prevented by the mucus and also bicarbonates. Mucus and bicarbonates. Okay? And then, next point. The HCl provides optimal pH. HCl provides optimal pH, that is 1.8 for action of pepsin. For action of pepsin, it also kills the microorganisms. It also kills the microorganisms. It also kills the microorganisms ingested along with food. Ingested along with food. Next. The proenzymes of gastric juice. Proenzymes of gastric juice. Gastric juice, pepsinogen and prorenin. The pepsinogen and prorenin on exposure to exposure to HCl or hydrochloric acid are converted into active enzymes like pepsin and renin respectively. Okay, next, Pepsin converts proteins into proteases and peptones. Pepsin converts proteins into proteases and peptones. Proteases and peptones. Renin is a proteolytic enzyme. Renin is proteolytic. Renin is a proteolytic enzyme. Proteolytic enzyme of gastric juice of gastric juice of infants. It acts on milk protein. It acts on milk protein. It acts on milk protein. The casein in the presence of calcium ions. The casein in the presence of calcium ions. Of calcium ions. And converts into calcium paracasinate and convert into calcium paracasinate and proteases Pepsin acts on calcium paracasinate. Pepsin acts on paracasinate and converts into peptones. Converts into peptones. Then prorenin, inactive enzyme, convert into the renin, active enzyme by HCl, pepsinogen, inactive, convert into pepsin, active form, and then casein is convert into paracasinate or calcium paracasinate. Calcium paracasinate plus proteases. Here renin plus calcium ions. Calcium paracasinate calcium paracasinate convert into peptones. Convert into peptones by pepsin. Proteins are converted. Proteins are converted. Proteases and peptones. And peptones by pepsin. This is about the digestion of proteins in the stomach, right? The next one is functions of liver. Functions of, what are the functions of liver? Next question is second, what are the functions of liver? The first function, liver secrete bile juice. Liver secrete bile juice. Bile juice, it contains bile salts, such as glycocollates, such as glycocollates, and tarocollates. Of sodium and potassium. Sodium and potassium that help in emulsification fats. That help in emulsification of fats. First point. And the second one is second on, liver plays a key role in carbohydrate metabolism. Liver plays a key role in carbohydrate metabolism. Carbohydrate metabolism, that is glycogenesis. One is glycogenesis, means conversion of glucose into glycogen. And next, glycogenolysis. Conversion of starch into glucose. Conversion of glycogen into glucose. And the next one is gluconeogenesis. Neogenesis. conversion of fats and proteins into glucose. That process is called the gluconeogenesis. And next, lipogenesis. Lipogenesis. Breakdown of the fats. Breakdown of fats. All these processes are their key role in liver, and liver also play a role in lipid metabolism. Next point, liver also plays a role in lipid metabolism. In lipid metabolism. In lipid metabolism. Synthesis of cholesterol and production of triglycerides. Synthesis of cholesterol and triglycerides. Next, the liver, deamination of proteins. Deamination of proteins. What is the deamination? Removal of NH2 group from the amino acids. A removal of NH2 group of amino acids. And conversion of ammonia and conversion of ammonia into urea. Via ornithine cycle. Next one. Lactic acid formed during anaerobic muscle contraction. The lactic acid formed during anaerobic muscle contraction. During anaerobic muscle contraction is converted into the glycogen. In liver by Cori cycle. Next, And liver is a chief organ of detoxification. Liver is chief organ of detoxification. Detoxification of toxic substances. Detoxification of toxic substances that enter the gut, enter the gut along with food, along with food, along with food. Okay, and liver acts as a thermoregulatory, acts as thermoregulatory organ like skeletal muscle and liver to take part in thermogenesis. To take part in thermogenesis, it has high glucose at its disposal. It has high glucose. It has high glucose at its disposal. At its disposal. And the liver acts as a hemopoietic organ. Liver acts as hemopoietic organ in the fetus. In the fetus. And erythroclastic organ in the adult. Erythroclastic organ in the adult. What is the destruction of the RBC? It is called erythroclastic. And hemopoietic means production of blood cells or blood. And next, liver synthesizes plasma proteins. Liver synthesizes the plasma proteins. Synthesizes plasma proteins such as albumin and globulin. ఒక్క 10 నిమిషాలు వచ్చేసేం స్ట్రీమింగ్ ఆపేది. And globulin, and also in the blood clotting factors. Blood clotting factors, fibrinogen and prothrombin. Fibrinogen and prothrombin. Wabinogen and prothrombin, etc. And anticoagulant called heparin and also produce anticoagulant heparin. Anticoagulant called heparin. That's all about the liver functions. Liver functions are also very important for IP point of view. Next one. The next chapter, breathing and exchange of gases. Breathing and exchange of gases. In breathing and exchange of gases, we can explain the process of inspiration and expiration under normal condition. This is the 4 marks question. Explain the process of inspiration and expiration. Inspiration and expiration under normal conditions. Under normal condition. That is the question. First, the breathing means maximizing the process of gases exchange. Breathing is means of maximizing the process of gases exchange. Maximizing the process of gases exchange. Gaseous exchange. It involves 2 stages. One is inspiration. Second one is expiration. First, inspiration. What is the inspiration? Intake of atmospheric air into lungs. Intake of atmospheric air. Intake of atmospheric air into lungs. Into the lungs is called inspiration. Is called inspiration. So inspiration, it is an active process. It is an active process. It is an active process as it takes place by contraction of as it takes place by the contraction of muscles of diaphragm and external and external inter external intercostal external intercostal muscles. The contraction of the diaphragm increases the volume. The contraction of diaphragm or diaphragm increases the volume of thoracic chamber, increases the volume of thoracic chamber, increases the volume of thoracic chamber in anterior-posterior axis, in anterior posterior axis. The contraction of external intercostal muscles. The contraction of external intercostal muscles lifts up the ribs and sternum. Ribs sub sternum, increasing the volume of thoracic chamber, increasing the volume of the thoracic chamber, dorsoventral axis, dorsoventral axis. The overall increasing the volume, thoracic volume, and increase the pulmonary volume, and increase the pulmonary volume. Then decrease intrapulmonary pressure, decrease intrapulmonary pressure, which forces the air, into lungs.